Okay, thank you very much. So it's time to go to the second speaker, Mahasi Shoman from Stanford University in Mike Schneider's lab. And now we will get into a bit of SARS-CoV-2 work.
Yeah.
So please, the floor is yours.
Thank you very much. Thank you, Mike, for inviting me. So I'm Mahasi Shoman, and today I'll be giving a talk on comparison of SARS-CoV-2 spike protein targets in mRNA versus inactivated vaccine and the associated autoantibodies. So a little bit about me. So during my graduate study, I worked with Dr. Joshua Lieber at Arizona State University, and there I have worked extensively on designing protein microarrays from scratch. So I used plastic microarrays, glass microarrays, and also silicon microarrays. And also I worked on various beads-based assay, which was popularized by Luminex. So we did on our own from scratch. So we used different functionalization technique, such as EDC-NHS chemistry. We used protein AG beads, and then we used biotin-streptavidin beads. So using this different chemical chemistry, we tried to functionalize the surface of the beads and use it for our immunoassay. And lastly, which like the DNA barcode assay, which is like popularized by Oli. So we had our own version, and it was like we used the same, the idea is same, but it's different in the way that we used barcodes and conjugated to our antibodies using click chemistry, and based on that, we identified that. So moving on, I also now work with Dr. Snyder at Stanford, and we use single cell B and T cell receptors, and I study the sequence of those and try to see how our immune dynamics change in various diseases. So using these different techniques, we try to understand how the disease progresses, what are the parameters which lead to the disease progression, how this immune system evolves due to infection, autoimmunity, or any cancer, and then identify various biomarkers for those diseases. So high throughput affinity-based techniques, these are already mentioned by different groups, so I will not go into much details about this. So when we started in the Josh Lab, so we really wanted to use high throughput techniques. So when I say high throughput, means we want to do the entire proteome. So as you can imagine, the proteomes for every organism is like thousands to even higher than that. So humans, like when we have like 10,000 human proteins, it's not easy to like purify, express all the proteins, purify it, and store it. So that's like a huge task, right? So we thought about it, and then we thought what we can do to like streamline the process and make it much more easier to do this scale of high throughput scanning. So what we did is we thought like instead of using proteins, which are much more sophisticated, what we want is we went one step back and we used plasmid instead of proteins. So as you know, like working with plasmids, we were working with DNA is much more easier compared to protein. So what we did is we cloned all the sequences, whatever proteins we have, we are interested about, cloned into a T7 PAN7 CGST vector, and then printed those plasmids into the microarray. And these microarrays are like very stable. You can keep it at like room temperature of 4 degrees, and then also use and express the proteins whenever you need using your in vitro expression kits. So this gave us really helpful in like increasing the scale to more than 10,000 proteins at a time. So as another speaker mentioned yesterday about the DNA ASU, so all these clones, we have currently more than 300,000 clones in the DNA ASU plasmid repository, and there is like human proteins, there is mouse proteins, there is different microbial proteins, so it's very easy to get those proteins and clone it according to like gateway cloning or any other cloning method to transfer to a vector which is useful for you. So we published our data based on this. We had like thousands of proteins, and then we probed against hundreds of human beings, and we checked like the autoantibodies which are commonly available in healthy individuals in cell reports in 2022. So some of the applications we did at our lab is we looked into, like as I mentioned, we looked into the antibody profile of a person. So this antibody profile of a person I looked into in this the cited paper, I looked into IBD patients. So IBD is a very interesting disease in the sense because our gut is like populated by trillions of microbes, and they are continuously interacting with our immune system. And this interaction is which regulates how our gut axis, gut-brain axis, and how inflammation occurs. So it's, as you can imagine, we have so many microbes and there are so many proteins. So it's not easy to just check those microbial proteins which can be used as a biomarker. So we use the same platform to check various microbial proteins we printed, and then we did discovery and then validated independently, and we found a very strong ROC curve which distinguished the people who had IBD and who didn't have IBD. We also looked into post-translational modification. We did glycosylation, we did citrullination on the slides, and then we identified with a specific antibody which binds to it, and then we found that we can demonstrate post-translational modification using microarrays. And then the epitope map I'll show you today in this presentation, and then also how you can check protein-protein interaction, protein-DNA interaction, so you can check various pathways, biological pathways which are being regulated, and also transcriptional factors. So today, the talk, I'll be talking about a cohort which we collected in, from one part is the individuals who got mRNA vaccine, and then the other part which got inactivated vaccine. And the mRNA vaccine was collected at California, and then the inactivated vaccine was collected at Shanghai by Dr. Shang Tao. So we collected samples it's a longitudinal study, and we collected the samples at 3 time points. So before dose 1, before they got the first vaccine, we collected the sample. After the dose 2, and post 6 months, we collected the samples. Similarly, we tried to make the same, get the same values, get the same time points from inactivated vaccine as well. So if we look into what are the differences, why do you want to look into this type of vaccines and why it's interesting? So these are the two, mRNA and inactivated is the most common vaccines used for SARS-CoV. And mRNA as is being mostly used in the Western countries, while inactivated vaccine is used mostly in the Eastern Far East, India, China, and parts of South America and Africa. So mRNA vaccine is a new technology, and it requires ultra-low temperature for transportation, which is commonly there in Western countries, but it's not available in all other countries. So that's why inactivated vaccine much more makes sense. The efficacy is really high for mRNA though, but if you see the modification, if there is a mutation, it's very easy to modify because it's just an mRNA molecule, while in comparison, inactivated takes a lot of steps to change. So why we are interested? So we looked into one of the paper where we saw that in the mRNA vaccine, there are 2 mutations. So one, the lysine has been changed to a proline, and the valine is also changed to a proline. So both the Moderna and Pfizer vaccine had these 2 mutations. So the reason for these mutations is that it stays in the pre-fusion conformation. So the mRNA vaccine stays in this position. When you check using electron microscopy, when you check the inactivated vaccine, so the inactivated vaccine was found to be in post-fusion conformation. So as you can see that there is a huge difference between both the structures, and that's why we thought there would be some differences in the targets of the spike protein which are being targeted. So before we move on, it's important to know whether our cohort, whether these individuals are just vaccinated or they have also got infected of natural infection. So when we collected the samples, we asked individuals, and most of them said they didn't get infected, but we had to verify it. And that's why we used a Luminex-based assay, and we checked using the common viruses, viral proteins, and tried to see whether they have antibodies to this. So if you see here, some of them have the EVP, which is very common in USA, and then also some of the influenza, which is a seasonal virus, so we expect it. But if you see for the SARS-CoV, most of the individuals don't have any infection, except for 3 or 4 which they got some natural infection. So we know like whatever we see will be due to vaccination and not because of some natural infection. So we did a peptide microarray. So what we did is we took the spike protein and we chopped the spike protein into small peptides of 12 mer peptides with sliding window of 6 mers. And then once we have these peptides, we synthesized using the peptide synthesizer and then printed on the microarray and probed it with the serum and tried to see which regions of the spike protein they are binding to. And we took the median of all individuals just to make sure that it's representative of that entire cohort and not just representative of some strong or very low signal. And if you look into the top part, so the top is inactivated vaccine epitope map, and the bottom one is the mRNA one. So if you see in the inactivated vaccine, the epitope map, the targets are mostly located in the RBD region. But when you look into the mRNA vaccine, the same peaks come up, but then there are some additional peaks which are there in between S1 and S2, and then hepatoid repeat 2 in the HR2 region. So this really was interesting for us, and what we anticipated, we found something similar to that. So we wanted the next thing was to check where these epitopes are located. So we used PyMol to check in the spike protein where the epitopes are located in the spike protein. And when you see this is the side view, you see these are the red ones are the highlighted epitopes. And as you can see, these are on the surface exposed epitopes. You look from the top view, you see that these are the ones which are the inactivated vaccine targets. And then the mRNA vaccine targets are mostly in the bottom part and on the sides. So this gives us the confirmation that whatever we are seeing on our peptide microarray is indeed are the very immunogenic regions, and they are surface exposed, which can be easily targeted by the antibody. Because we have this like longitudinal samples, so we wanted to check how this antibody level changes over time. Does it increases, decreases? How does it do? So we looked into the top 5 immunogenic region of the spike proteins. And we plotted this for the top 5 regions of the spike protein in inactivated vaccine, and plotted for those two, and then post 6 months, what is the level. And we checked the average of the top 5 regions, and we found that there is a 2.24 times depletion in antibody level over time. So we did the same thing just to see in also mRNA vaccine. And to our surprise, it reduces by even more. It reduces by 5.86 times in mRNA vaccine compared to the inactivated vaccine, which is 2.3 or something like that. So there is 2-fold more decrease in antibody level in the mRNA vaccine. So even though the mRNA vaccine initial response is much more stronger, but then it also depletes very fast compared to the inactivated vaccine. Because when we took the samples, we asked the individuals self-reporting whether they have some symptoms or not. And so we wanted to check why some individuals have side effects and why others don't have. And that's why we divided the group into 2 different groups, the individuals with side effect, individuals without side effect. And we see that their targets are same, but they have much more stronger immune response, individuals with side effect. So this shows that the individuals with side effect have much more sensitive immune system, and that's why they have less tolerance for this, and they get a much more stronger signal. These antibodies which are generated are often linked to long COVID disease. So we have people reporting symptoms which last for months after COVID vaccination or COVID infection. So we wanted to see which are the pathways they regulate, and is there any relation to those? So we looked into the autoimmune part to understand if there is any autoimmunity pathways which are being activated in these individuals. So we probed 16,000 human proteins against the plasma of the parties, and this is using CDI who brought microarrays. And we tried to check what is there at the baseline, like the pre-dose one, and then what are the antibodies, autoantibodies which shows up at post-dose. And as you can see here, we have the IgM and IgG. So these are the target antigens against which autoantibodies increased in after dose 2. So there was nothing in between. After vaccination, these autoantibodies increased. Similarly for IgG, these are the targets, and as you can see, the scale IgG is expected to be much more stronger than the IgM, and that's what we see. So I was curious whether these are somehow similar to the spike protein, and that's why I did a multiple sequence alignment of these antigens with the spike protein to see if there is any sequence similarity between those. And I found that most of them don't have like exact match. It is just 3, 4 amino acid and then some gaps. But we see that there is no extraordinary linear epitope match. So we believe that these epitopes are most likely conformational epitopes. So these are the antibodies which was initially originated for the spike protein, SARS-CoV protein, and then it cross-reacted to one of the human proteins, or one of these human proteins which have similar 3D conformation. And that can explain why we see these autoantibodies after the second dose. So we also looked into divided this into two groups. So we have these individuals who had side effect and who don't have side effect. So we wanted to look which pathways they regulate, and is there any biological pathway we can say. So we looked into the antibody targets, which are the autoantibodies which elevated in the individuals with symptoms and which didn't elevate in those individuals who didn't have symptoms, and check those target antigens and see what they do, what are the functions they do. Using gene ontology analysis, we found that they actually regulate a lot of them regulate biological regulations, cellular processes, and biological metabolic processes. So this can explain why the most common side effect we see, like muscle ache and fatigue, can be regulated by these biological pathways. So moving on, we also looked into the B-cell repeater sequences. So what we did is we checked all the BCR sequences, and then using 10x genomics, we checked what are the BCR sequences in those individuals. So these all circles, one circle represent one clone, which is like same V gene, same J gene, and CDR3 sequence. So before dose 1, if you see here, so these is the clonal repeater of the cohort, and these are some of the clones which are shared clones which are like expanded. When you look into post-dose 2, there has been an expansion of this clonal expansion of some of the clones. And then again at 6 months after that, it again goes back to its original state. So as we expect that after vaccination, our immune repeater expands and generates a lot of antibodies, and that's what we see here. So we looked into this, specifically these clones, and tried to see what these clones are exactly. And then we did a B-cell lineage map of these expanded clones and did a phylogenetic trees of these clones and tried to see which are the antibody sequences this represent. So we got the germline sequence, and from these germline sequences, you can see the highlighted ones are the sequences which have undergone a lot of somatic hypermutations, and these are the sequence of the antibody which are due to this SARS-CoV vaccination. So we have the sequence, and we know exactly which these antibodies are. So key takeaways of my talk. So inactivated vaccine targets mostly in the RBD region, while mRNA vaccine targets in RBD as well as other parts of the SARS-CoV protein. The immunogenic regions, whatever we found, are all exposed in the surface of the spike protein. And then the humoral immunity, what you see in mRNA vaccine, depletes twice as fast as the inactivated vaccine. Several autoantibodies we found which were elevated after post-dose 2, which are most likely the conformational epitopes which are similar to SARS-CoV spike protein. Gene ontology revealed the pathways they most likely regulate, and that's why we see the symptoms in those individuals, the side effect in those individuals. And then BCR analysis revealed that there is a clonal expansion after the vaccination, and the antibody sequence of those expanded clones were revealed. This is my last 30 seconds. So this is my last slide. So a lot of students here, even I also always think about whether you should go for affinity-based or mass spec. So as you can see here, there are some advantages of each of this technique, and we have specificity is one of the problem with, Olink is doing pretty good job, but I think specificity is one of the problem with the affinity-based techniques. Mass spec is very high. Expertise is relatively low in affinity-based, while mass spec is pretty high. Throughput, both of them have improved a lot, and both of them are pretty high. And then cost is relatively low, and then mass spectrometry is high because of the machine cost. Quantitativeness, more of them, they are relative and then absolute. So one of the main is like the advantage of mass spec is if you don't know the protein, still you can analyze it, but this affinity based is dependent on you should have the antibody for it. So what I think is both of them complements each other, and there is no for some of your techniques maybe affinity based is better, for some of them mass spec is better. So I think this type of conferences helps to push the limit of both these techniques, and it will be really helpful for more advancing the field of affinity based. With that, I would like to thank and take questions.
Thank you so much. I think there's a question coming up. Please go ahead.
From me?
Oh.
Hi, thanks for the talk.
Yeah.
So I was wondering, there have been a couple of reports in the literature recently that mRNA vaccines push the immune response to IgG4. Is that something you have looked at all or could comment based on your findings here?
Can you repeat the last part? I didn't.
So if you get individuals that were vaccinated with mRNA vaccines, you have a very high proportion of IgG4 antibodies.
IgG4?
Yeah. Is that something you looked at? Or
So we didn't look into all this specific isotopes, so we just look into IgG as a whole. So yeah.
Okay. Really interesting to check. Thanks.
Yeah, thank you.
Andrew?
So, you said the mRNA vaccine is pre-fusion, the inactivated vaccine is post-fusion.
Yes.
So you're assuming that the differences between the responses are due to the structure of the protein, but it could also be due to the form of the mRNA versus protein. How do you extract the
yeah, so the structure is the different structure is reported in one of the other paper which I cited. So that's not what we did. But the antibody, we see that there is difference in the regions of these in mRNA and inactivated vaccine, but that doesn't say that one should be stronger than the other. So it's just that inherently, I think mRNA initially leads to much more stronger because there is only the spike protein is being released. That's why initially the antibody level is much more stronger. But then in inactivated vaccine, because they are all the part of the virus, so the initial strength is not that high, but eventually it levels up.
No, so you have two variables. One is the form of the vaccine, and the other is the structure of the protein against which you're vaccinating, right?
Yes.
And you have, and both. You're not. Do you have any information as to which of the variables are important? Is the difference the difference in the response you're seeing, is it due to the form of the vaccine, or is it due to the form of the structure in the two vaccines? Do you have any idea?
I think it's not because of structure, the difference in the level. It's more of how these vaccines are so different. So the mRNA vaccine, how it targets only. So it's not just the structure, it's how inherently how they are so different.
So also a very interesting study, but I wonder whether you considered that the mRNA, which is used for transcription or translation of the spike protein, is not, in most cases, not completely transcribed or translated.
Mm-hmm.
In other words, you have only fragments of the spike protein, so can this be the reason for this weak response or faster meeting response towards mRNA vaccination?
Yeah, I think that's a very important insight, and I feel that can be one of the reasons why the strength initially is higher, and because there is no some of the it's a fragment of the protein and not the complete protein, and that's why it goes away and fades away faster. yeah, that's a good insight.
Yeah.
So, as far as I understand, the response that you're measuring is to linear epitopes, is that correct?
So,
The response that you're measuring is to linear peptides.
yes, yes.
So, what fraction of the response do you think you're actually capturing with this assay? And how do you know that any of the responses that you're measuring are neutralizing?
So, interesting question. So, we don't like, in this study, we don't do the neutralization assay. So our collaborator, like Shang Chetao, he did the like separate like neutralization assay of these epitopes and checked which epitope is much more neutralizing. So we didn't do any neutralization assay in this study. Yeah, and then for the peptide, whichever we have, like these are the linear epitopes, and because this is a peptide microarray, so yeah.
Well, we know that most of the antibodies, at least to the prefusion, are conformational, so you're not going to be able to detect those. And so I don't really know what this study actually means.
Yeah, I mean, so that is a good question. So I think our goal here is, because the targets we are checking, because if you like peptide microarray, again, it can't be like conformational because there's a limitation of what we can do. So we really wanted to compare, and that's why I think we used the peptide microarray and not like 3D conformational. So it will be difficult for us to like check where it's binding using 3D conformation.
And just one final question. When you compared the individuals with and without side effects, how many were they? And did you look into the individual variation?
Yeah, so for that we have like around 20 and 20 individuals. So
20?
20, but some of them have like different side effects. So some of them muscle ache, some of them have dizziness, some of them have fatigue. So we didn't divide them into individual side effects. Means that would be a good way if we have a big cohort, but then if you divide them, it will be very small. The cohort size and the statistical significance will be very less, so we didn't do like divide them into individual groups. But that is something we were always thinking about, yeah.
Okay, thank you very much.
Yeah.
Thank you so much. And this session is thereby completed.